Updates, agency updates. Would Dr. Wharton please lead?
Good morning. I'll be presenting the CDC agency update. At the December 2019 meeting of the Board of Scientific Counselors of the Infectious Disease Centers, the Deputy Director for Infectious Diseases and the Vaccine Confidence Work Group was established to identify gaps in CDC activities to increase vaccine coverage, inform the center's efforts to provide recommendations to CDC on the agency's strategy to strengthen vaccine confidence and maintain high vaccine coverage at the national, state, tribal, and local levels. Over the next 2 years, the work group will collect and analyze relevant information and provide findings and observations to the BSC of the infectious disease centers for deliberation and potential votes. The Vaccine Confidence Work Group will be chaired by external experts, will be supported by staff from across CDC, including the Immunization Services Division, the Immunization Safety Office, and the Division of Viral Diseases. For measles during 2019, a total of 1,282 individual cases were confirmed in 31 states. In October, New York announced the end of the state's nearly year-long outbreak of measles. With that declaration, the United States was able to maintain its measles elimination status. The Pan American Health Organization has established a regional verification commission, and CDC will provide evidence regarding the sustainability of measles elimination. In December, American Samoa declared a measles outbreak. CDC has provided technical guidance on case and contact investigations, infection control, vaccine acquisition and usage, and mass vaccination efforts. We also provided American Samoa with additional funding to support their response to the outbreak and purchase additional doses of MMR vaccine. We heard yesterday a comprehensive update on the influenza vaccine season. I would just like to add a couple of brief updates that I don't believe were included. We do track we track influenza vaccine coverage weekly through the National Immunization Survey and are able to provide estimates that are updated weekly. And I'm pleased to see that this year coverage appears to be tracking a little above last year. It's currently around 60%, and if it continues to increase, we will likely end up with a higher coverage than we had at the end of the season last year. We're also pleased to see an increase in doses of pediatric influenza vaccine ordered by states for use in the Vaccines for Children program. Obviously, vaccine availability doesn't necessarily mean we'll have higher coverage, but we can't have higher coverage if there aren't more doses out there. And so we're pleased to see that increase in doses ordered for next year for the VFC program. Finally, the 2020 National Adult and Influenza Immunization Survey annual in-person meeting will be held on May 18th, 2020 at the Westin Peachtree Plaza Hotel in Atlanta, Georgia, to be followed by CDC's National Immunization Conference, which will be May 19th through 21st, also at the Westin Peachtree Plaza Hotel in Atlanta, Georgia, and information about both of those meetings is available online. Thank you.
Yes. Now, Dr. Allen, update.
Hi, good morning, everyone. My name is Dr. Kara Elam, and I am the Research and Policy Strategist for the Office of Infectious Disease and HIV/AIDS Policy, OIDP. I will be giving a brief update on OIDP's immunization-related activities, as well as updating you on the National Vaccine Advisory Committee, NVAC. The last NVAC meeting occurred February 13th through the 14th and focused on vaccine confidence and innovation. The agenda included presentations on recent research in vaccine innovation, updates related to the coronavirus outbreak, and efforts to advance the 2019 Executive Order on modernizing influenza vaccines in the U.S. The agenda also included sessions on challenges and opportunities for combating mass misinformation online with speakers from large social media companies such as Twitter, and the use of storytelling to increase vaccination awareness and rates. The ASH in a prior meeting charged NVAC to develop and formally submit reports with implementable recommendations for vaccine confidence and immunization equity. During the meeting, each of these subcommittees provided an update on their progress and preparations for drafting and submitting these reports on time. The next public NVAC meeting will take place June 9th through the 10th in DC. If you cannot attend in person, there will be a live webcast and information for this upcoming meeting, and past meetings can be found on OIDP's website. The National Vaccine Plan. OIDP will be releasing the 2020 National Vaccine Plan late this year. At the last ACIP meeting, Dr. Tammy Beckham, OIDP's director, updated this committee on the progress related to the development of the 2020 National Vaccine Plan. The work on this plan is ongoing and includes review and incorporation of feedback solicited through public comment and stakeholder discussions that took place October of last year. This consisted of 26 one-hour discussions with individuals and a variety of stakeholder groups, including healthcare providers, industry, academia, and patient advisory organizations. Much of the feedback received aligned closely with previous NVAC recommendations, including aspects such as a goal for innovation and including a global goal as relevant to the ongoing coronavirus outbreak. At present, we are integrating the feedback received into a concise, actionable 5-year plan that will provide vaccine strategies across the life course, guide priority actions for 2020 through 2025, and identify indicators to measure progress toward plan goals. As will be emphasized in the next National Vaccine Plan, vaccine innovation continues to be a priority at HHS as we recognize the critical role of innovation within the U.S. vaccine and immunization enterprise. OIDP is putting innovation into practice in multiple ways. For example, in order to improve pertussis vaccine performance, we convened scientific, policy, and regulatory experts to discuss ethics, feasibility, and regulatory considerations of a controlled human challenge model. This human challenge model will provide a better understanding of the immune system's response to infection, including colonization, and guide pertussis vaccine development in the U.S. As opposed to current practice, the challenge model will entail colonization as the desired endpoint. The output of this meeting was instructive and will serve to guide future policy. A white paper will be published in spring of this year. As you know, HPV remains a significant public health challenge with vaccination completion rates for adolescents barely above 50%. The ASH has prioritized increasing HPV vaccination rates nationally in order to reduce vaccine-preventable cancers. As a result, OIDP is implementing innovative approaches to significantly improve HPV vaccination rates within the next 5 years. This is being achieved through a number of activities, including working with the CDC, the American Medical Group Association, and the American Cancer Society's National HPV Vaccination Roundtable to establish a learning collaborative among integrated delivery networks and other large health systems. Additionally, we have launched the innovative Million Cancer Preventing Congregations program, a faith-based initiative that empowers leaders to participate in prioritizing HPV vaccination and cancer prevention within their congregation. In addition, I would also like to provide an update on 2 national plans that will be released later this year. First, we are updating the National Viral Hepatitis Action Plan. The plan, which was first released in 2011, provides goals, strategies, and indicators to achieve a coordinated national response to hepatitis A, B, and C infections. The updated plan is scheduled to be released this summer and will prioritize available hepatitis vaccination. OIDP is also advancing the first ever STI Federal Action Plan. Recent data shows that rates of STIs have grown considerably over the past 5 years and have reached an all-time high in 2018. Acknowledging this serious public health concern, HHS is developing the STI Federal Action Plan to guide a strategic federal response. This plan will focus on the 4 most common STIs that have the greatest public health impact in the U.S. chlamydia, gonorrhea, syphilis, and HPV, where HPV vaccination will be emphasized as a cancer-preventing measure. The plan will focus on cross-cutting issues, including stigma, disparities, and social inequities. In closing, I would like to say thank you for the opportunity to provide this overview.
Thank you very much,
Dr. Mullock from NIH.
Good morning. I have a few brief updates for you. First, I wanted to make sure you're aware that NIAID is supporting the development of a number of COVID-19 vaccine candidates, both by intramural and extramural investigators. We're looking at multiple approaches, including vaccines based on technologies that have shown promise against coronaviruses that cause SARS and MERS. The Vaccine Research Center at NIAID is collaborating with the company Moderna on the development of a vaccine candidate using a messenger RNA vaccine platform expressing a recombinant spike protein of SARS-CoV-2. We anticipate that this experimental vaccine will be ready for clinical testing in the next few months. We will continue to conduct our preclinical studies as well as the first-in-human study of this candidate and other candidates. We are coordinating closely with our colleagues at CDC, HHS, the Food and Drug Administration, Department of Defense, and other federal partners. Second, on February 26th, NIH announced the start of a randomized controlled clinical trial to evaluate the safety and efficacy of the investigational antiviral remdesivir in hospitalized adults diagnosed with coronavirus disease 2019 at the University of Nebraska Medical Center in Omaha. This is the first clinical trial in the U.S. to evaluate an experimental treatment for COVID-19. The first trial participant is an American who was repatriated after being quarantined on the Diamond Princess cruise ship that docked in Yokohama, Japan, and volunteered to participate in the study. The study can be adapted to evaluate additional investigative treatments and to enroll participants at other sites in the U.S. and worldwide. And third, as Nancy Messonnier mentioned yesterday, CDC had received a clinical specimen of the first U.S. patient infected with SARS-CoV-2 and prepared the sample and then sent it to the NIH BEI repository. And that sample has been grown up and is available to investigators to use in their research endeavors for those who are registered. We are providing additional materials for coronavirus research, and those materials are being updated on a daily basis as we get more information. And you can go to the BEI website for more information about those resources. Thank you so much.
Thank you. HRSA, Dr. Rubin.
Good morning. Thank you for allowing me to provide this update. First, I'd like to circle back about Ebola vaccine coverage by the Countermeasures Injury Compensation Program. Based on the current declaration, the Ebola vaccine is a covered countermeasure, and injury claims that are from the Ebola vaccine are eligible for benefits as long as they meet program requirements. And as of January 1, 2020, the Countermeasures Injury Compensation Program has compensated 39 claims totaling $5.5 million. Now, the National Vaccine Injury Compensation Program has continued to process an increased number of claims. In fiscal year 2019, 1,282 claims were filed with the VAERS. In that same fiscal year, $196.2 million was awarded to petitioners, and $29.2 million was awarded in attorney's fees and costs. This includes fees for compensated, dismissed, and interim cases. As of January 1, 2020, 289 claims were filed with the program, and $57.2 million was awarded to petitioners and attorneys for attorney's fees and costs. As of January 1, HRSA has a backlog of 913 claims alleging vaccine injury awaiting for review. More data about the program can be obtained at our website. Thank you for the update.
Thank you very much. Dr. Fink, FDA.
Good morning. First and foremost, FDA is engaging with vaccine manufacturers, other federal agencies, and regulatory and public health authorities across the world to facilitate development of vaccine candidates to address the current COVID-19 outbreak. Second of all, since the last ACIP meeting, we've had 4 major vaccine approvals, the first of which you heard a lot about yesterday. That's Ervebo, the VSV vectored Ebola vaccine that was approved in December. We have had 3 influenza vaccine approvals, 2 of which were to add quadrivalent formulations to previously approved trivalent seasonal influenza vaccines. This includes a quadrivalent formulation of Fluzone high dose, indicated for use in individuals 65 years of age and older. That was approved in November. And then a quadrivalent formulation of Fluad, MF59 adjuvanted influenza vaccine that was approved at the end of January, also for use in individuals 65 years of age and older. The fourth approval was for an adjuvanted H5N1 vaccine for positioning in the Strategic National Stockpile. This is an MF59 adjuvanted vaccine manufactured using the same platform as Flucelvax, a licensed seasonal influenza vaccine. The trade name is Audens, and this vaccine was studied for safety in nearly 4,000 individuals, which included over 300 pediatric subjects and nearly 1,800 individuals 65 years of age and older. The effectiveness was inferred by hemagglutinin inhibiting antibody responses post-vaccination, and also inferred from efficacy data with Flucelvax, the vaccine manufactured using the same platform. This H5N1 vaccine was approved under our traditional approval pathway for adults 18 years of age and older, because Flucelvax is approved under that pathway as well. For pediatric individuals under the age of 18, Flucelvax is either approved under accelerated approval provisions or not yet approved, as is the case for individuals less than 4 years of age. And so once the ongoing studies to support traditional approval of Flucelvax are completed, then Audens will serve as the basis for full approval of Audens in those pediatric age groups.
Thank you very much. Department of Defense, Dr. Dusing.
Hi, good morning. The Department of Defense appreciates the ACIP and CDC's continued inclusion of the DoD in this meeting and the ACIP working groups. I'd like to share the following brief update from the DoD. We're closing out our 2019-2020 influenza vaccination campaign after reaching our annual goal of vaccinating over 90% of personnel by January 15th. This included active duty, select reserve forces, and healthcare providers. New for the 2020-21 season, the DoD will introduce Southern Hemisphere influenza vaccine to support operational activities in applicable regions. The DoD, in coordination with other agencies, is moving forward with limited implementation of the newly licensed Bavarian Nordic smallpox and monkeypox vaccine. The Immunization Healthcare Division is developing a protocol to evaluate this vaccine for cardiac safety, interaction with other vaccines, and also optimum booster interval. I've spoken about the pragmatic assessment of influenza vaccine effectiveness in the DoD, or PAVED study, before, and it's now in its second research year. Over 5,800 patients were enrolled this year at 9 different research sites, and this study group is co-chaired by the Immunization Healthcare Division and the Uniformed Services University Infectious Disease Clinical Research Program, or IDCRP. And I look forward to giving updates on its progress at future ACIP meetings. The South Atlantic and Pacific Regional Vaccine Safety Hubs continue to actively present research on a number of subjects, including atypical shoulder pain and dysfunction following immunization, vaccine hesitancy in the military system, effects of anti-inflammatories and analgesics post-immunization. Finally, in fiscal year 2019, the Immunization Healthcare Division responded to over 1,400 calls to its call center, reviewed 1,022 VAERS cases, and reviewed 408 clinical cases, and conducted 322 on-site quality assurance surveys to ensure the highest standards of care throughout the Department of Defense. Thank you for your time.
Thank you. Dr. Weiser, IHS.
Good morning. Thank you for the opportunity to address the committee. The Indian Health Service would like to report our influenza vaccine coverage for this year so far as of February 10th. We have given 267,565 doses of influenza vaccine with a population coverage among children 6 months to 17 years of about 36.1%, and among adults 18 years and older, 33.6%. In addition to this, IHS facilities have a mandatory healthcare personnel influenza policy, and interim influenza vaccine coverage among federal IHS healthcare personnel was 94.8% as of December 31st. One of the software updates that we are grateful to be anticipating will be established soon is the final phase of new immunization forecasting software, which will finally allow us to catch up with some of our newer vaccine recommendations. We expect this to go live in the next 30 to 60 days. And then finally, addressing vaccine hesitancy. The Northwest Portland Area Indian Health Board Tribal Epidemiology Center, with funding from CDC, is working with various stakeholders, parents, community members, healthcare providers, and a community organization called Boost Oregon to develop approaches and communication strategies to strengthen conversations among healthcare providers and community members around the importance and benefits of childhood vaccines, and to reduce vaccine hesitancy within tribal communities. The project's aims are to educate patients and parents about childhood vaccines, increase healthcare providers' confidence and ability to discuss patient and parent concerns about vaccines, and to encourage providers to strongly recommend immunizing all children according to the ACIP published vaccination schedules for children and adolescents. Thank you.
Thank you. Dr. Kim at TVA.
Good morning. I have 4 announcements for the Department of Veterans Affairs. The first is that VA updated its clinical preventive services guidance and national electronic decision support tools, which are also known as clinical reminders, for human papillomavirus and pneumococcal immunizations based on the most recent ACIP recommendations. Located on the VA internal website for clinicians, the VA guidance also includes public-facing links and resources for staff to use with their patients. Second, we have a seasonal influenza update from August 1st, 2019 through February 1st, 2020. Over 1.9 million flu vaccinations were administered within VA facilities. This does not include vaccines received outside VA. In addition, more than 109,000 enrolled veterans received a no-cost flu vaccine through a partnership with Walgreens. VA Notice 2020-02 was released to the field on the storage of vaccines and medications in pharmaceutical-grade purpose-built refrigerators and freezers at VA medical facilities. And lastly, VA public-facing Staying Healthy recommendation charts for men and women have been updated based on the CDC 2020 adult immunization schedule, and they're available on the VA National Center for Health Promotion and Disease Prevention website at www.prevention.va.gov. Thank you.
Thank you. We'll now move on to other topics. So first topic of today is rabies vaccine with presentations by Dr. Fry, Rao, and Blanton. And Dr. Fry will give us an introduction, please. What?
Good morning. We're going to start today talking about the activities of the rabies work group. Before we start, I would like to introduce you once again to the work group members, which include Lynn Abata and Jose Romero, and also our CDC leads, who include Agam Rao and Jesse Blanton. I'd like to thank also our liaison representatives and invited consultants, and also our CDC technical work group and subject matter experts. I'd like to review the terms of reference. This is 2 slides. Our terms of reference were to determine the epidemiology and the burden of rabies exposure and PrEP and PEP administration in the United States. Evaluate and revise recommendations as needed for the vaccination schedules, route, and site of PrEP and PEP administration. To consider evidence that was generated to inform the rabies recommendations of other global organizations such as WHO. Review rabies exposure risk and risk assessment guidelines for the general population and by occupational and recreational groups. We are also asked to evaluate serological and monitoring recommendations, including whether recommendations should differ depending on the degree of rabies risk for a person and whether adequate antibody titers are needed to confirm immunization. Consider whether any recommendations should differ for healthy adults compared to immunocompromised persons, children, and pregnant women. Update recommendations with information about the 2 rabies immune globulin products approved by the U.S. FDA during 2018, and to identify areas in need of further research for informing future vaccine and immunoglobulin recommendations. What we went over during the last meeting included the following. We provided background, including epidemiology, clinical course, and rabies biologic in the U.S. We discussed factors that contribute to decisions for pre-exposure prophylaxis, or PrEP, and post-exposure prophylaxis, PEP. We discussed the role of neutralizing antibodies as a marker for adequate vaccine response, and we introduced PrEP schedule considerations, which included immunogenicity and duration of immunity, route of vaccine administration, role of booster doses for select populations, and reasons for PrEP failures. Since we saw you last, we concluded data review and discussions about PrEP topics and began discussing PEP topics to include PEP schedules, data about rabies immunoglobulin or RIG administration, and presentations about newly licensed RIG products. So for today, we plan to review background to PrEP, vaccine safety, and working group considerations, rabies vaccine schedule and duration of immunity, including the systematic review data that we looked at, and other guidance and next steps. Our new tentative timeline is the following. In June, we hope to provide PrEP grade and ETR, evidence to recommendation, and some PEP data. And in October, we hope to have a PrEP vote and also continue to discuss PEP data, including grade and evidence to recommendation. And by February 2021, we hope to have the PEP vote. Thank you.
Thank you. Dr. Rao, please.
Thank you, Dr. Fry. Rabies in humans is an acute progressive encephalomyelitis that is nearly always fatal. It is transmitted from infected mammals by a bite, scratch, or exposure to saliva or neural tissue. It is not transmitted by exposures to blood, urine, or feces of infected animals. There have been no known laboratory-confirmed cases of human-to-human transmission through contact with infected persons. In the U.S., there have been 0 to 4 cases per year. There are few animal species that are reservoirs for rabies. Rabies virus variants, or RVV, are named for animal reservoir species in which they circulate. They're confined to geographically definable regions, which we'll see from our next slide. Infection can be transmitted from the reservoir species to other species. For example, a raccoon RVV can spread from a raccoon to a cat to a human. So it's important that it be known that RVV does not denote the animal to which the human was exposed. Within the U.S., canine RVV has been successfully eliminated. We're left with terrestrial or wildlife rabies and non-terrestrial rabies. Terrestrial or wildlife rabies is rabies for which the reservoir is wildlife, like skunk, fox, mongoose, and raccoon. And the map to your right is the distribution of terrestrial RVV in the U.S. Non-terrestrial rabies is rabies where the reservoir is bats. Bat RVV is widespread, present in 49 of 50 states in the U.S., all but Hawaii. Activities that have led to confirmed cases in the U.S. are the ones that bring humans closer to rabid animals or rabies virus. They occur in domestic and international settings. In both of those settings, recreational and occupational work like laboratory and field work have been involved. Domestically, there are also potential exposures, including contacts in everyday life, which occurs if there are bats in the home or residences in a wooded area with opportunities for exposure to rabid animals. There have been rare transmissions from organ and tissue transplants, too. So for these reasons, preventing rabies is a priority. Vaccinating domestic and wild animals is one way of doing that. The USDA and health departments and others work on oral rabies vaccine campaigns for this purpose. Vaccinations in human, pre-exposure prophylaxis or PrEP, and post-exposure prophylaxis or PEP have been found to be highly effective in preventing rabies and likely contributing to the low number of cases that we have in the U.S. Pre-exposure prophylaxis is what we're talking about today. It's comprised of a rabies vaccine schedule, and over 15,000 people receive PrEP per year in the U.S. So I'm going to talk, since the PrEP schedule involves rabies vaccines, I want to tell you a little bit about them. There are 2 rabies vaccines currently licensed in the U.S. There's a human diploid cell vaccine, or HDCV, and a purified chick embryo cell vaccine, or PCECV. The brand name and the manufacturer are listed in the second and third columns, and as you can see from the last column, both vaccines are licensed in the U.S. for intramuscular administration only. Both vaccines have been around for decades, and no concerns were raised during the 2008 ACIP recommendations. However, because these vaccines are an important part of PrEP and PEP regimens, the work group did look at the available safety data. The work group looked at passively collected data from the Vaccine Adverse Event Reporting System, or VAERS. Data for HDCV is listed on this slide. There were 1,666 reports received during 1990 to 2019, and the take-home point is that the majority, or 94%, were non-serious. Systemic reactions were observed as shown here on this slide, but serious adverse events like angioedema were rarely reported. These findings are consistent with pre-licensure and post-marketing studies. For PCECV, VAERS received 739 reports, and here as well, most, 93%, were non-serious. The most common systemic reactions are listed in the second bullet, but these findings, too, are consistent with findings of pre-licensure studies, not new information. To ensure that we had evaluated any published data about safety since the 2008 ACIP recommendations, we did review the literature for trials, and we found 25 papers, the details of which are listed on this slide in the sub-bullets of the first bullet. But in the end, the findings were consistent with what's been previously reported. We found no changes to the safety profile. So who should receive rabies pre-exposure prophylaxis? The purpose of PrEP is to provide partial immunity to persons who are at risk for rabies. It's not a substitute for PEP. Persons who receive PrEP still need PEP after an exposure. Rabies immune globulin wouldn't be needed for these people, and the vaccine series would be shorter, but receiving PrEP does not negate the need for PEP. It's intended for persons who may have unrecognized or frequent exposures to rabies virus. For persons who have a delay in starting PEP, for example, if a person traveling internationally is in a rural area and they have to travel to the capital city in order to receive PEP, then PrEP might be a bridge until they have access to PEP. The recommendations for PrEP vary depending on the level of risk to unrecognized exposures. Risk categories for PrEP were developed with this in mind. They were developed to outline the populations that should receive PrEP based on a shared risk level, and then provide corresponding recommendations for schedules and frequency of titer checks that are specific for that level of risk. Risk levels were named for the level of risk for unrecognized exposures, where continuous is the highest risk, followed by frequent and infrequent, and finally rare, which is the least risk. The rare category is the general U.S. population. This is the PrEP guide from the 2008 ACIP rabies recommendations. I'm just going to walk you through the information in this slide because all of our presentations today are based on understanding this table. If we focus only on the left side of this slide first, the first column is the risk category. Starting at the bottom is the rare risk. There's increasing risk for inapparent or unrecognized exposures as we move up on the table. to the continuous category. And then the second column, the nature of risk, describes features of each risk category. For example, the nature of risk to unrecognized exposures is the highest for the continuous risk category. Persons who fall into this risk are exposed to rabies virus in high concentrations, and frequently they can be exposed from bites, non-bites, or aerosol exposure. The nature of this risk decreases in intensity as you go down the second column. So for the general population in the rare category, the exposure is always episodic, and if it occurs, and the source is recognized. The recommended schedule, as I mentioned on the previous slide, and the frequency of titer checks differs depending on the risk category, and that's why these risk categories are very important. The information in the third column, the typical population column, is what frontline providers use to identify the category in which their patient falls so that they can follow across and determine the PrEP recommendations. The populations described in that third column depend on several factors. They depend on the activity performed by the population, the geographic region of the mammal that they might be exposed to, and access to safe and effective PrEP. And I'll talk about each one of those in the next few slides. So by activity performed, we mean occupational and recreational activities. Occupational exposures include jobs that require handling live rabies virus in diagnostic, research, or vaccine production capacities. For example, a rabies diagnostician. Some occupations have a lot of contact with rabies and some with terrestrial mammals. As part of recreation, too, some people have exposures that are risky for rabies. People can have contact with bats if they're a spelunker, and people also travel internationally and could potentially have exposure to rabid animals. Geographic region is important because different regions have different risks regardless of the occupation and recreational activity that a person might be doing. For example, domestically, a wildlife worker in Washington state, so the top left of the map where no terrestrial rabies virus variant is noted, may involve a lower risk than the same worker in Pennsylvania, which is deep in the green zone here in Raccoon RVV. Similarly, a spelunker entering only caves in Hawaii, where there are no rabies at all, may have a lower risk than a spelunker who enters caves in Alabama, where there is. Internationally, the important surveillance information is knowing whether canine RVV is an issue. Geographic location is not the only important consideration for travelers. Access to PrEP is a very important issue too. Nearly all major cities have rabies vaccine and rabies immune globulin internationally, but if a person is, for example, doing recreational activities in a rural area, they may not have quick and easy access to the PrEP that is in the major city, and there may be a delay in their receiving PrEP in those cases. So this table in the 2008 ACIP recommendations intends, through this third column, the typical populations column, to account for these issues. However, over the years, we've heard that this column can be hard to follow. This is a closer look at the contents of that column. On this slide is the typical populations column to the right. To the left of the typical populations column is the corresponding risk category. Some of the reasons this column was confusing to folks was that, for example, veterinarians are listed in two different categories, and it wasn't clear on immediate review of the table what the differences were. It does state that the geographic regions are different for these types of veterinarians, but it was still difficult to follow. It mentions here in the frequent category all persons who frequently handle bats. People didn't understand the reasoning for why it was in the frequent versus infrequent category. And then travelers in the infrequent category said travelers visiting any area where rabies is common and immediate access to appropriate medical care, including biologics, is limited. The content is all there, but was found to be challenging for providers who didn't find their specific occupation of the patient in front of them to be listed. And since the application of the ACIP recommendations in the last column of that table is dependent on the clarity here, the work group made some changes. So we split the contents of that population slide into two columns, which are shown here, the disease biogeography and the typical population. I'll walk you through the information in these columns by risk factors in the subsequent slides. Understanding these categories is important to understanding the next two presentations, which is why I'm going to spend a little bit more time here. One column is for disease biogeography, and the second column in the red box is for the population. In the row for the continuous risk category, the disease biogeography is the laboratory, and the typical population is personnel who work with live rabies virus in research, diagnostic, or vaccine production capacities. There's also examples listed here that the work group elaborated on, necropsy, so that the provider has a better sense of who all might fall into that category and make the best decision about a patient whose job is listed. Necropsy of a suspect rabid animal or working with rabies virus cultures are listed. Obviously, this population is at the greatest risk of rabies exposure, and the PrEP recommendations reflect that. For the frequent risk category, there are two different populations that have a high enough risk of rabies to be included here. Both are based on the principle that if you have frequent exposure to rabies, you fall into this category. So in geographic regions where bats are the only reservoir for rabies, anyone who frequently handles bats would have frequent exposure. An example would be a bat biologist. And then in geographic regions where terrestrial mammals are reservoir for rabies, anyone who works with these terrestrial animals are at frequent risk. Those populations would include animal care professionals like veterinarians, veterinary technicians, animal control officers, and others who repeatedly handle terrestrial reservoir species like wildlife biologists, rehabilitators, and trappers. This is exactly what we see here in the first row. People can have professions that are similar to those in the frequent category, but be listed in the infrequent category. That's what you see here, animal care professionals, others who repeatedly handle terrestrial reservoirs. Spelunkers was added to this row. This was previously in the frequent category, but the work group felt that the recommendations for those in the infrequent category might be a better fit. The second row here in the infrequent category includes geographic regions where terrestrial mammals are the reservoirs for rabies. The population is different from that of the previous slide in that these veterinary students have less exposure than a veterinarian would. It also includes short-term volunteers and people for whom maybe frequency of titer checks would not be as important as those who would fall into the frequent category. All right, so the third row here is the geographic regions internationally with canine rabies. Travelers will be performing activities would put them at increased risk for exposure to rabid dogs and may have difficulty getting access to safe PrEP, for example, in rural areas. Children may receive PrEP depending on the country to which they will travel. And this all aligns with the soon-to-be updated CDC Travelers Health Destination pages and the 2022 Yellow Book as well. So these are minor changes that were made to the table that are expected to clarify the contents and make it more user-friendly. The information, that last column about PrEP schedules and the frequency of serologic titer checks is really the subject predominantly of the next presentation, but I'm going to speak a little bit about it in the next few slides. We did make a few other changes to this for clinical guidance. The minimum antibody titer level was changed to 0.5 IU/ml to ensure standardization with WHO and the labs nationwide. And then also the work group recognized that the whole disease biogeography aspect, because it was not its own column, may created some confusion about the importance that should be placed on it, and therefore highlighted the importance for state health departments to be contacted if there's any confusion about those issues. So vaccine series and frequency of titer checks. Intradermal or ID rabies vaccine administration has been globally recommended since the 1980s. WHO, as recently as their 2019 recommendations, recommended ID because of its dose and cost sparing. And for that population, the population the WHO guidelines are for, dose and cost sparing recommendations could make the difference between a person getting a vaccine and not getting the vaccine, so potentially life or death. For For the U.S. population, though, it's a different population. In the U.S., rabies vaccine is not licensed for ID use. It is packaged for one intramuscular injection. There's less of a need to vaccinate many people concurrently, as there might be in developing countries. And so the risk of infection that could come from multiple punctures to a vial that is intended to be a single-use vial, and therefore without preservatives, might not outweigh the benefits. Most of the populations for whom PrEP is recommended in the U.S. are because of the work performed in their occupations, so presumably occupations would pay the cost of these, and so for that reason, the work group focused on intramuscular. The 2008 ACIP PrEP schedule is intramuscular rabies vaccine on day 0, 7, and day 21 or 28. This is the schedule for all persons in the continuous, frequent, and infrequent risk categories. The frequency of titer checks, however, differed depending upon the risk category. It was every 6 months for those with the greatest risk to unrecognized exposures, and it was every 2 years for those in the frequent risk category. And for those in the infrequent risk category, the guidance was that titers are not needed. And then, of course, no PrEP, and therefore no titer checks were recommended in the 2008 ACIP recommendations for those in the rare category. Rabies antibody titer is used as an indicator of adequate immune response to vaccine, but acceptable titer is not an indication of protection. It's a surrogate. A person with a low titer may still be immune. That person might very well still mount an anamnestic response if they had a rabies exposure. But because of the high mortality associated with rabies and the high risk for some populations, guidance has always been to try to maintain a higher than the minimum acceptable antibody titer for these people at their routine titer checks. A lower titer might still have protected them, but a sustained high titer is preferred. So given all of this, the work group's task was to evaluate whether the vaccine series could be different depending upon the risk category by looking at data for a 2-dose IM series and data about a booster for those in the higher risk categories. And secondly, to evaluate whether frequency of titer checks could be different depending upon the risk exposure category by looking at data that helps determine whether titer checks for persons in the continuous category should be more often than those in the frequent category. And this data will be the subject of the next presentation by Dr. Blanton. And so with that, I want to thank all of our work group members and our CDC team, and I think I can take questions.
Open for questions. Dr. Hunter.
Could you go back to the proposed table and walk me through what you would do for a bat biologist in an area that had terrestrial, geographic regions where terrestrial mammals are reservoirs for rabies?
So that's, can you see this okay?
No.
Okay. So that would, anyone who is a bat biologist, if they're practicing in the United States, for 49 out of 50 states, they would fall in that frequent risk category, the first gray box. So geographic regions where bats are the only reservoirs for rabies, persons who frequently handle or come into contact with bats. I see what you mean, and probably the word only is not needed there, but where bats are the reservoirs for rabies, they would fall into the frequent category.
Thank you. I think removing the word only would be advisable.
We'll do that. Thank you.
Dr. Bell.
Thank you. I was curious, I was interested to see your estimate that 15,000 people a year receive pre-exposure prophylaxis, which struck me, not based on anything, I guess, as being kind of a lot. And I was curious about whether you had any information about what comprises the largest proportion of this group of people. Is it international travelers, or, because a lot of these people seems like there wouldn't be that much of a turnover that you would be vaccinating that many people every year.
Yeah, I don't know the exact, I think most of them are occupationally associated, but we do know that among these occupations, veterinarians, for example, have to get vaccinated as part of their schooling. They're required during their veterinary schooling to get vaccinated. But some of these other professions, when there are manuscripts published about this, we find that they're not as good about, I guess, getting vaccinated and maintaining those titer checks if they require it. So we assume that it's the highest categories that are the ones that are accounting for the most number, and not as many travelers, probably.
Dr. Rockwell. No, sorry, Dr. Sanchez.
I just have a question. With respect to the indirect exposure, I mean, it just educated me. I was not aware that rabies was droplet spread. Or what situations? Would that be during intubation process?
It's droplet exposures. There have been situations where laboratorians have, through manipulation of the virus, there's been aerosolization of the virus, and that's what we're referring to. So the infrequent folks, if they're just getting a bite or a scratch, that would be a direct exposure, whereas those who are kind of manipulating the virus might be at risk in other ways.
But in a clinical situation, it would not be droplet.
In a clinical situation, yeah, I guess it would be droplet, actually. We do worry about people who've had droplet exposure through intubation. But the pre-exposure prophylaxis is not indicated for clinicians, and there has never been any transmissions that have occurred in the clinical environment. But that is part of the the post-exposure risk assessment that is done, the type of exposure that someone may have had to a case that eventually becomes confirmed for rabies.
Dr. Freihofer.
Sandra Freihofer, American Medical Association. This is a question about possible exposure. So there are some very rural parts of Georgia, and from time to time I have patients that are hunters come into my office. You mentioned the trappers being at risk, but a lot of times trappers send things to taxidermists, and then taxidermists, they make things that people put on their walls. And is there any risk past the trapper to rabies?
Well, I suppose if it was soon after the animal died and there was saliva present on its nails or saliva present somewhere, then it could be a possible exposure, but the risk would probably decrease once time had elapsed since the animal died.
But you've never heard of any kind of exposure related to that?
I have not. I don't
That
know.
is somewhat
Brett, have you
reassuring
heard of anything like
to the
that?
hunters.
Dr. Fry.
I don't know what a taxidermist does with the guts, the brains and spinal cord, for example. I guess it's not close enough. I don't know what a taxidermist does, how long a taxidermist waits to manipulate a dead animal and what it does with the neural tissue, the brain and the spinal cord. I don't know at what point in time that's manipulated.
Yeah,
So
I guess
I
I'll
can't
defer
answer.
to Brett Peterson and see if he has any thoughts on that.
There's always the possibility that there could be transmission of rabies virus if there is saliva or other infectious materials like neural tissue in contact with mucous membranes or any break in the dermal barrier. So if that is documented, then that would be indication for post-exposure prophylaxis. However, at the same time, the rabies virus is not a very hardy virus. It can be killed by desiccation as well as UV light. So depending on the state of the carcass, you could potentially determine the risk based on those factors. If it's dry, it's more than likely not a high risk. But all of those kinds of risk assessments can be done on a case-by-case basis.
And that would be mostly post-exposure versus pre-exposure, too.
Thank you. Other questions or comments? Very good. Thank you very much for that presentation. Dr. Blanton?
Thank you, everyone. I have the opportunity to talk a little bit about some of the systematic review that we've been working on in the work group. And just kind of as a way of orientation, I'll first be talking kind of the broader systematic review activity that was done, and then focusing specifically on the meta-analysis that was done around the 2-dose vaccination series. We conducted this review looking at the immunologic response of to rabies pre-exposure prophylaxis, looking primarily at the primary response, the duration of immunogenicity, and the response to booster vaccination. This systematic review was actually started in 2017 and then using the same protocol for the working group updated through 2019. Our population was very broad. We were looking at any persons that were exposed with the potential for rabies exposure, and looking primarily at persons as alternates, looking at persons who received an alternate rabies vaccination schedule, and secondarily looking at rabies vaccination by alternate routes, primarily intradermal, for comparison. And these were all, of course, being compared to the gold standard ACIP recommendations, which is the 3-dose, 3 to 4 week schedule by the intramuscular route. And with our outcomes being focused on rabies neutralizing antibody, and again looking at 1 to 3 weeks after vaccination for the primary period, at 1 year post-vaccination, and following booster vaccination. We examined multiple databases, a period from 1965 to 2019, and this really represents the period in which modern rabies vaccines were either under study or available. And after searching, resulted in a little over 250 unique papers. Our exclusion criteria were to exclude papers that used older nervous tissue vaccines or experimental vaccines, and we were only looking at immunocompetent populations. And again, looking at pre-exposure prophylaxis with a broad definition of that, essentially any persons receiving 1 to 3 doses of vaccine over any form of schedule. We were looking at papers that provided information on immunological response to vaccination measured by the RIFIT, which is the gold standard neutralization assay, and those studies that reported out using either geometric mean titers or seroconversions at a stated cutoff, and in this case, across all papers, that cutoff was the WHO recognized 0.5 IU per mil. So the little more than 250 papers after screening and critical review, we resulted in 63 publications total, ranging from 1978 to 2019, a total of 146 study arms or cohorts, and I'll talk a little bit more about that later, and over 11,000 subjects that were involved in the studies. These were all largely clinical trials of one form or another, conducted mostly in Asia and North America. And as I mentioned earlier, we had a broad representation of schedules that were studied ranging from single dose, 2-dose schedules from the 1 week 0-7 all the way to a 2-month schedule, and multiple 3-dose schedules as well, again from a 1-week schedule to the current recommended 3-dose 3 to 4 week schedule. Multiple vaccine types were used, the PCEC and HDCV vaccines that are licensed in the U.S., as well as the PVRV, which is a virocell vaccine that has a broad utilization internationally, and primarily intramuscular and intradermal routes. And just broadly for this overall systematic review, what we found looking at the general schedules is that from a primary vaccination response, there really wasn't much of a difference. There was no significant difference in the seroconversion rate between these different schedules, and the working group really focused on the 1 week 0-7 from a practical standpoint, a practice of being a shorter period to administer the vaccination series, as well as being concurrent with the new WHO recommendations. So before I get into that, just to give you a quick broad perspective of what the meta-analysis of the existing 3-dose 3 to 4 week schedule looks like. As we mentioned in the October meeting, it's a very well established, more than 40 years experience with this schedule. There's a very high seroconversion after vaccination with the 3-dose schedule. And really just lines up when you look at the seroconversion rate 1 to 2 weeks after vaccination with near 100% seroconversion. I want to show a little bit of the comparison for the geometric mean titer for this schedule, because one thing that we found is that when we looked at these studies is that there's a considerable amount of heterogeneity or variability between studies for the geometric mean titer for vaccination. Here looking at differences between intradermal, these are all the 3-dose 3 to 4 week schedule, between intradermal and intramuscular studies. The intradermal is certainly significantly lower than the intramuscular, but the relative clinical difference between these, between the significant difference is somewhat questionable. While the seroconversion at 0.5 IU per mil is not necessarily a protective level, it is recognition that that's an adequate immune response, and that a person who has an adequate immune response and can receive a booster is generally protected against rabies challenge. So while we're not looking at the intradermal schedule, currently there are some intradermal studies that were utilized in the later analysis because we didn't want to turn that data away in an area where there are relatively few studies for some of these other schedules. So you will see some intradermal cohorts or study arms showing up in the later analysis. And I'll point those out as we go along. So moving to the rabies pre-exposure prophylaxis, this is the 2-dose 1-week schedule, day 0 and 7, looking at primary response. Of our larger systematic review, there were 12 total studies that provided information that could be used for the pooled analysis for the 2-dose schedule. And one of the things that we had to do is because these clinical trials really didn't have any sort of direct ratio comparison internally, we actually found that we could break these study arms apart and treat them as more of an observational cohort for this pooled analysis. And so that's generally how while these original studies were controlled trials, we treated the individual study arms as from more of an observational perspective. So that allowed us to take out from these studies some of the individual study arms that either were not the schedule that we were interested in, or for the purposes today, removing vaccines that are not licensed in the U.S. For the analysis. We also, for the primary study, and one of the other advantages of looking at the 0-7 schedule is that the 0-7 schedule is the first 2/3 of the 3-dose schedule. And so for studies that can provide information and serological data on day 14, 21, 28, before the 3rd dose is received, is information that we can look at for what the primary response is to those first 2 doses of vaccine. The last 2 studies that are highlighted in orange, Sochins and Indy et al., are the 2 studies that looked exclusively at comparisons of the 2-dose vaccination schedule to the 3-dose, and those will be highlighted throughout the rest of the presentation for reference if you're trying to make comparisons. So first, looking at the serological response to the geometric mean titer, this is again individuals who have received only 2 doses of vaccine at day 0 and 7. Looking at the differences here in the top group are persons who had serology taken at day 7 to 14 after the 2nd dose of vaccine. The second grouping are those 3 weeks after the vaccination. And you can see that there is kinetics growth in the antibody response between these groups. And it's important also to point out that there are a relatively limited number of studies, similar to what was presented earlier for the 3-dose schedule, quite a bit of heterogeneity presented between these studies, but there is a robust response moving up to nearly 10 IU per mil within 1 to 2 weeks after vaccination, and 12 in the subsequent week. Perhaps more directly comparison is, again, looking at the seroconversion rates, which we see very consistent results in reporting across the different schedules. Again, 1 to 2 weeks after the 2nd dose and 3 weeks on the bottom. And there is essentially nearly universal seroconversion between 7 to 14 days after the 2nd dose of vaccination. And this is really more of our apples to apples comparison. We're now looking at 30 to 60 days post-vaccination. So for the top group, these are individuals who have received the full 3-dose schedule, 0, 7, 21, or 28. And the bottom group is our individuals who received only 2 doses on day 0 and 7. And from a seroconversion standpoint, we see 100% seroconversion in these groups 1 to 2 months post-vaccination. Again, the limitation here being the small number of studies that were available for some of these analyses. So moving from the primary vaccination, we'll look a little bit at the data that was available for duration of immunogenicity and the response to booster. Same as before for the tables, there were 13 studies that were available that had information available for this analysis. removing some of the individual cohorts that were using non-U.S. licensed vaccines. And we'll look at this to beginning at the response in individuals 1 year post-vaccination. 3-dose vaccination groups are on the top, the 2-dose vaccination on the bottom. And you can see that there actually is no significant difference between the 3-dose and 2-dose schedules when we look at the geometric mean titer 1 year out. However, when we now look at the information of 1 to 2 weeks after the booster dose, so this is 1 year after vaccination, a booster received, and now serology taken 1 to 2 weeks after that booster, we do actually see that there was a significantly higher anamnestic response in the 3-dose group versus the 2-dose 0-7 schedule. Again, both of these are high above the sort of minimal IU per mil, 0.5 IU per mil, and the difference here again is somewhat questionable what the clinical significance would be. Shifting this to the seroconversion rate, we actually see the opposite scenario here, where the individuals who received 1 year post-vaccination, again 3-dose on top, 2-dose on bottom, we actually see that the 2-dose group had a more rapid decay in the number of individuals with an adequate immune response. About 60% of individuals that received 2 doses still had an adequate response at 1 year, compared to 90% amongst those who had received 3 doses. However, administering that booster dose, we again see a complete universal anamnestic response in all individuals, boosting above that minimal level. So just to summarize these studies individually, when we look at the 2 primary studies that provided information on the serological response, Sochins et al., this study, while it was administered entirely intradermal, it was one of the larger sample sizes. They found that amongst the individuals that had a pre-booster serology taken in a 1 to 3 year window, that the 2-dose intradermal group generally had a significantly higher GMT compared to those who received 3 doses intradermally. But again, as seen with the other data, 100% of these groups had an adequate titer after booster. Similarly, India et al. Actually presented data on both 3-dose and 2-dose by intramuscular and intradermal. Similarly, found no significant difference. Unfortunately, this study was a little bit smaller sample size in the study arms, but found no significant difference observed in the GMT at 1 year for either 2-dose, either intramuscular or intradermal. Did find a lower number of recipients with an adequate immune response at 1 year, but all recipients had an adequate titer after receiving a booster at 1 year. Looking back out again a little bit at the larger systematic review, because there's really more, given the time that the 3-dose 3 to 4 week schedule has been around, there's been more time to actually look at the long-term kinetics with this series. And in general, the studies have looked at it, and this has been found on shorter time periods with some other 2-dose 0 and 28-day schedules. But have generally found that there's a rapid decay, regardless, over the first 6 months post-vaccination that slows to around 1 year. The decay has been found to be more rapid when administered by the intradermal route. But more importantly, the studies have generally found that in situations where a booster dose is administered, and typically it's given around 1 year in most of these studies, that there's a greater increase in titer than what was observed in the original primary response amongst individuals, and that there's a slower decay after booster is received. And we could actually see this with some of the systematic review meta-analysis, where we look at individuals that do not receive a booster dose at 2 years, and generally find that only about 85% are reported with an adequate titer at that time period. We've seen this very largely in cohorts of veterinary students, compared to individuals who do receive a titer at 1 year and tend to maintain do receive a booster at 1 year and tend to retain much higher adequate antibody rates in excess of 95% over long periods, up to 4 or 5 years, in some of these studies that were conducted over longer time periods. So this is some of the information that we're looking at in relation to the 2-dose schedule and in relation to consideration of recommendations around boosters for higher risk groups that allow us to look at extending some of the serological monitoring periods for these different risk groups. Again, thanking the rabies vaccine working group. Thank you.
Dr. Hunter.
So my question is going to be, could you consider the first dose of a post-exposure prophylaxis as the booster for a 2-dose pre-exposure prophylaxis series? And let me, I'm not sure you're going to be able to answer that from your area of expertise, but if there are more people who are involved with the public health response and clinical response could respond to this. The way I'm thinking about it is first of all, you need to be in a situation where you should get the pre-exposure prophylaxis, have ongoing exposures. And then you need to be able to recognize that you had an exposure. And then you need to be in a place where you can get, you're not out in the bush and you can get the post-exposure prophylaxis. And then I think my main question is, how long do you have from the time you recognize an exposure to when you need that amnestic response? And are you going to get enough from the first dose of your post-exposure prophylaxis in time to protect you if you had a reasonable exposure? Does that make sense? Okay.
Okay, I guess I'll try to answer that one. So I think what I'm hearing is you receive your primary course, then you go off somewhere into, let's say, international travel out in the bush somewhere, and you have an exposure that you're aware of. That's the first thing. Are you aware of it? And then you seek follow-up. The incubation period for rabies is a little bit complex and rather broad. The estimates are broad. And the idea is that once you've had an exposure, you would seek immediate attention as soon as possible. If you're out in the bush, that's difficult to do because it may be weeks or longer until you can get aid. And then the question is, when you go seek aid, is there product available to vaccinate? And so you're asking, can you give, is the post-exposure dose enough to boost the pre-exposure prophylaxis? It depends on what the period of time has been and how quickly you can get it, whether it's going to be appropriate or not. So I can't answer that question. If you can get there right away, it would be great. I think if you're going in a situation where you can't get those kinds of that kind of care, you have to think differently about your pre-exposure prophylaxis.
Dr. Bell.
Yes, thank you. I always find these deliberations about the schedule for a vaccine like the rabies vaccine where the mortality is pretty much 100% and you don't have enough cases, any way to evaluate efficacy to be very challenging. And so this idea of, shortening the schedule, again, I remember when we went from 4 doses to 3 doses, again, I think is a little bit challenging. And I guess the question that I'm wondering about, if you have any sense of, is it seems like the issue of getting boosters for people that have gotten pre-exposure prophylaxis and you want to keep their titer in some, quote, safe zone, and you don't want them to miss their boosters, perhaps becomes more important to continue to have this kind of margin of safety for people who have some sort of ongoing exposure. So I guess this is like a coverage question that, as I say, you probably don't have any data about, but I'd be interested in your at least anecdotal sense of the extent to which these populations that are covered with the existing pre-exposure prophylaxis recommendations, in fact, are compliant with getting the recommended booster doses.
Yeah, so getting at the exact question of who's getting the booster dose is a little bit more difficult. We do know that looking at information on compliance around things like serological monitoring we have some of the information there. We know generally there's not great compliance. So we know that there's already large segments of the veterinary population that are probably the best tracked that are not getting serological monitoring over these time periods. And that have populations that are probably 20-30% that are no longer meet the minimum titer level that they would be recommended to receive a booster at that level. And so this is sort of the anecdotal of that. We've not had any ever reported cases of failure in those populations.
Dr. Talbot.
Yep, thank you. It looks like from slide 14 that this is really being based on the study with 22 patients. And that is a little concerning to me because that's a very small number. And as Dr. Bell pointed out, this is a fatal disease. I think this is one of those instances where we know the vaccine is safe and that you could recruit 200 people and give them 2 doses and follow them over a period of time and collect some really nice data. And it doesn't have to be an elaborate study. And I think along those lines, I think it's time that we have the data on how many of our vets in high risk are getting repeat vaccinations in serologic. And I think that's a study that can be done. We may have to work with the Veterinary Association or other groups, but I think this is data that's needed for us to make a decision because this is a 100% fatal disease.
You have a comment, Dr. Fry?
Sorry. Thank you for those comments. I think there's a lot of studies that could or should be done in this field to answer those kinds of questions. I know for the laboratorians, for example, they are routinely checked, I believe it's every 2 years, and their levels are maintained above 0.5 international units per ml. And the veterinarians are fairly good about having their titers checked, but I think there are some who aren't as good as others about getting their titers checked. And they're the ones who I think might be at risk also for unknown exposures as well as the laboratorians.
Dr. Bernstein?
So I was wondering whether, so it seemed that after 50 years of literature that you were talking about, I thought there were some instances where one vaccine might be preferred over the other. If I understood or what you were saying, that certainly either vaccine would, as a booster, 100% response either way, but it sounded like duration was better with the IM than with the intradermal. And I didn't know whether that would, people would prefer to use an IM rather than an intradermal initially.
So it's a really good question, and it's one that there's been quite a bit of research looking around that. Generally, there, as mentioned, the intradermal route tends to have a little bit slower decay than the intramuscular over long periods of time. But again, what the relative clinical implications of that are very much questioned of what it's statistically significant what that implication is as far as protection isn't well understood. While we know that more is better, depending on the type of exposure and contact that you have. The difference between intradermal and intramuscular is probably negligible in the difference of that decay. For the booster effect, it tends to be, again, both of them have a very solid anamnestic booster. for that reaction. And the intradermal route's been used pretty extensively throughout the rest of the world, and no real difference in failure rates or anything like that has ever been observed with the intradermal route. A lot of the issues around it come down to the practical side of having enough throughput in your clinics and issues like that to be able to reasonably use vials in a timely amount.
Thank you.
Dr. Talbot, follow-up.
Yeah, I just want to follow up to Dr. Fry. I do think these studies do need to be done, though, because if you're asking us to reduce the number of shots, we need to have real data. And I am not comfortable changing a schedule for 100% fatal disease when we don't have the data.
Okay. Well, does anybody else have further comments about that, or?
Dr. Atmar.
I agree with Dr. Talbot. I mean, I would be concerned about the paucity of data for the IM route, which is essentially what we're talking about in the U.S. and the recommendations we're making. I think it's likely that the ID data will translate over, it does translate over, but the fact of the matter is, from the data that we've been presented, it looks like there are considerably less than 100 persons from whom the data, looking at the primary responses that we have available for. So that doesn't really give us a lot of confidence about what the actual or real rates may be in terms of seroresponse or the GMTs.
Dr. Bell.
Yeah, I have to say that I also have a certain level of discomfort with the idea of shortening a schedule which saves a dose of vaccine for approximately 15,000 people a year, essentially where we really, to say, well, it's probably okay in this kind of situation, I feel a little bit uncomfortable with that. Also in a situation where it seems like, as people are saying, the compliance with these booster doses is really not great. And what we're concerned about is, one veterinarian, with a cat that had an unrecognized exposure to a raccoon. And, 2 months later, that veterinarian has rabies. I mean, that's kind of what you don't want to see happening. So I sort of feel like I would like to understand the rationale for doing this in the, in a situation where the potential adverse consequences are large.
Dr. Hunter.
I'm just wondering if, to follow up on that, if the ACIP will be faced with a cost-effectiveness analysis that is similar to what ACIP had to face with meningococcal B vaccine, which is a very severe sequela without much data because of the low frequency of the disease.
So let's move on to Dr. Rao's presentation, and then we'll come back.
Can I just make
Yes.
one more comment? So we are talking about a 6-month booster shot in some populations, 6 months to 12 months, and we'll hear more about that.
Dr. Rao.
Okay, thanks. So this is just a reminder about the table that we discussed during the first presentation that I gave, and the changes that will be made. I will change the issue with the only bats section, but the additions, just to remind you, are the splitting of the typical population part into 2 different columns. Also, the minimal acceptable antibody titer level at 0.5 IUs per ml is a change that aligns with WHO recommendations and is more of a standardized will be more standardized now. And then we're also highlighting the need for health departments to be consulted whenever there's any confusion at all about whether a patient falls into one category or another because there's uncertainty about the local epidemiology. And then this last column is the one that that would be based on data about whether the schedule and the frequency of the serologic titer checks would be changed. So that would be either not changed or changed depending on if there's data. All right, so the work group thought about whether that last column, the data about the schedule and the frequency of titer checks, would need to be changed for special populations. And so even though we don't know whether those recommendations would change, we wanted to consider what might be different about those patients. So the work group looked at data about the effectiveness of rabies PrEP for children, and children actually exhibited a very robust response, even better than that of healthy adults. So we don't anticipate there would need to be any special considerations for children than from healthy adults. For persons with altered immunity, studies found varying immune responses, and as mentioned in ACIP's altered immunocompetence guidelines, effectiveness, but not safety, is of concern for persons who are administered the rabies vaccine. The 2008 ACIP recommendations actually specifically state that patients who are immunosuppressed by disease or medications should postpone pre-exposure vaccinations and consider avoiding activities for which rabies PrEP is indicated. And when that's not possible, immunosuppressed patients who are at risk for exposure to rabies should be vaccinated and their virus-neutralizing antibody titers checked. So that would be something that's different from the healthy patients, and that a titer is not routinely checked after completion of a PrEP series for healthy adults, but for persons with altered immunity, it is something that the guidance is provided in the ACIP 2008 recommendations to ensure response. Now, for pregnant persons, there is data about effectiveness about PEP in that population, but not PrEP. The current ACIP recommendation is that if the risk for exposure to rabies is substantial, then PrEP might be indicated during pregnancy, is the language in the 2008 recommendations. The work group's preference was to state that there's no safety or effectiveness concerns for PEP administration in pregnancy, and that the decision to perform activities that would necessitate receiving PrEP during pregnancy is an individual decision. So that is something that would be added to from a guidance standpoint if the committee is in agreement, but there would be no changes made to the immunocompromised recommendation or guidance. Okay, so consideration for travelers. One of the things that we discussed on the work group was the co-administration of rabies vaccine with malaria prophylaxis. So in the 1980s, there was a report of a Peace Corps worker who received ID PrEP and chloroquine and was later bitten by a dog but did not receive PEP and ended up passing away from rabies. Her antibody levels at the time of her eventual hospitalization were found to be low, raising the question of whether antimalarials interfere with immunity from rabies vaccine. And the 2008 recommendations actually lumped in antimalarials within a sentence that included immunosuppressive agents by stating that corticosteroids, immunosuppressive agents, antimalarials can all interfere with the development of active immunity after vaccination. And although there wasn't any specific guidance for what to do if this were the case with antimalarials, the paragraph did go on to make the statements about immunocompromised states that I read on a previous slide. We, as a work group, reviewed a 2019 study that was recently published in the Journal of Infectious Diseases. And in that study, they compared ACIP's PEP-IM schedule after initiation of chloroquine, malarone, and doxycycline, so not just chloroquine as an antimalarial. And they found that co-administration of malarone and doxycycline actually resulted in high antibody titers. But for the arm involving chloroquine, the antibody levels were lower, but they were still 4 times higher than the minimum antibody cutoffs, and so possibly not clinically significant. The work group's preference then, based on this data, was to remove antimalarials from the sentence about immunocompromising conditions, since all antimalarials do not have an effect on active immunity, and instead possibly include a sentence that titers may be considered for persons vaccinated while concurrently taking chloroquine, regardless of whether the chloroquine is for antimalarial prophylaxis purposes. Okay, I'll move on to next steps now. These things are, I guess, not necessarily the next steps given the discussion that we just had for the last presentation, but I'll mention some of them. So we had, as a work group, come up with a policy question, which I think we're now potentially revisiting, but we were, for that first policy question, proposing should a 2-dose pre-exposure prophylaxis series involving HDCV or PCECV IM 0-7 days replace the 3-dose series IM 0-7, 21, 28 days for all those for whom rabies vaccine PrEP is recommended. And as you can see here, the population was persons for whom rabies vaccine PrEP is recommended. The intervention was the 0-7 days rabies vaccine IM PrEP schedule. Comparison was the 0-7, 21, 28 days schedule. And the outcome, there was only one outcome, and that was going to be immunogenicity, because the safety isn't a concern, as I mentioned earlier. And then we had proposed a second policy question, which was, should an IM booster dose of rabies vaccine be recommended 6 to 12 months after the 2-dose PrEP series IM 0-7 days for those in the continuous and the frequent categories of people who receive PrEP? And again, that would be laboratorians or animal care workers or people who work with bats, but animal care workers who work in terrestrial regions. And the population would be persons in those two categories for whom rabies vaccine PrEP is recommended. The intervention is 6 to 12 months rabies vaccine booster after 0 to 7 day IM rabies PrEP schedule. The comparison would be no rabies vaccine booster after 0 to 7 days rabies vaccine PrEP schedule. And the outcome was the duration of that immunogenicity. And again, safety was not planned to be an outcome, given there are no concerns about that. So that is what we had thought we were going to do, and I think this planning for the next couple slides might be amended here. But what we had thought was that we would complete the PrEP presentations in preparation for a vote at the June ACIP meeting, presenting the grading of the recommendations if there was going to be that change, and the evidence to recommend. And that we'd also begin PEP data presentations, provide background presentations about PEP considerations and data to inform clinical guidance and recommendations about the PEP vaccine series. The newly licensed rabies immune globulin products, there's been 2 since the last ACIP recommendations. The location of rig administration, WHO has made different recommendations than what the current ACIP recommendations say, and so we're considering that data. And then whether there needs to be any amendments to whatever is provided as recommendations or clinical guidance about PEP, whether there needs to be any changes or considerations for special populations. So for future ACIP meetings, beyond the June ACIP meeting, we had thought to address clinical guidance about handling deviations in PrEP and PEP, clinical guidance about managing the completion of PEP after a series is initiated internationally, like when a U.S. citizen travels internationally and then comes back to the U.S. after already having received a dose abroad. And then we've also taken the advice of the committee at the last meeting and are taking the text of the ACIP recommendations and trying to create a flow chart depicting the factors that providers should consider when deciding whether PEP is indicated. I know that I believe Dr. Hunter might have mentioned it at the last meeting, that there are online state health departments sometimes have some flowcharts or other systems in place to facilitate that decision-making by the physician. And if we can depict that in the ACIP recommendations, it could be helpful as well. And so we are seeking to do that. And there is like a fine line, because we can't provide all of the local epidemiology information that is necessary to make decisions, but we're trying to do so while keeping it still general. And then at some point during these meetings, obviously there will be votes for the PrEP and the PEP policy questions if there are policy changes. And that is all. I can
Dr.
take
Fry,
questions.
additional comment? Oh. Sorry, Dr. Adler.
I may misremember this, but, and I was always confused about the 2 levels of seroprotective titer. But my recollection was that 5 IU per mil was what was used previously, and in the WHO, 0.5 IU per mil. And while I understand the desire for alignment, I guess the rationale for the higher level being used up to this point, up to this change, and also the implications it might have for interpretation, for example, of the chloroquine data, where if you used a higher threshold, maybe there would be greater or lesser seroprotection after primary series. So can you remind us of what the prior threshold used was and what the data are to, other than the desire to align with WHO these many years later? Thank you.
So the recommendation as it stands now is complete neutral, 1:5 complete neutralization at 1:5, which actually ends up becoming less than 0.5 IUs per ml. And so by adopting a 0.5 IUs per ml, it's actually a higher titer level. And the populations that would be affected would be people at the highest risk category. So it wouldn't be a massive number of people, but it would be the people, the laboratorians, the people who are the animal care workers who are working in areas of terrestrial rabies or bat rabies. And it would be a higher titer, so potentially better.
Dr. Hunter.
What if the policy question were 0-7 and then 21 days to a year, anywhere in that range for the 3rd shot? Is that, what I'm not saying is, what the clinical implications of that, but how much would that mess up your timeline and your analysis?
Would you be suggesting that for only the people in the highest risk categories or for people lower down, like the international travelers?
All of them is what I was thinking.
Yeah. So for one of the reasons that the work group had thought about, we had earlier in our thought process thought that maybe we would keep the 0-7-21 for the highest risk categories, but we would drop down to a 0-7 schedule for the lower risk categories. The reason being that we know from the data that 0-7, for someone who's going who's going to have short-term summer volunteer work experience with animals, maybe the duration of a high titer is less important, and the 0-7 schedule would suffice. But it was starting to get a little bit hairy, like if someone ends up being in the lower risk category and then changes professions and is in the higher risk category, what do we recommend for those populations? And so it started to become a little hairy, but we had thought about whether the schedules might be different for different categories of people. And part of the thought process for this booster dose being okay was that the people in the highest risk categories are people who their occupations really should be enforcing it, like laboratorians, veterinarians, wildlife workers. You would think that there would be a occupational health clinic that is ensuring that these people get that 3rd dose 6 to 12 months later. And so that was our thought. I mean, similar to them getting the 3rd dose at 21, 28 days, trying to ensure that they get it 6 to 12 months later, we thought wouldn't be too much of a problem, and it wasn't needed for the lower category of people. But we hadn't thought about what you had said about 21 days to 1 year. We hadn't thought about that. For travelers, we know that the whole 21 days issue it creates some problems because people don't think beforehand about getting the entire vaccine series, and it might not be needed.
Dr. Fry.
One of the problems with travelers is that they wait until it's almost too late to get their full series in for their vaccination. And so that was one of the considerations with this 0-7-day vaccination schedule, was that they could accomplish that better than this 07/21/28 day. I think there was a little bit of data that showed that the 07/21 day antibody titer response was a little bit better than the 07/28, but it's not really anything that's significant. Again, for the high-risk, continuous risk folks, the idea is to keep that level, measurable level, at 0.5 international units per ml. That's a very nice level. It's thought to be protective. We don't really know what the lowest level of protection really is, but we know that that level is protective. And so we try to keep it up there. Any boost further out, 6 months, a year, gives a very nice increase in geometric titers.
Dr. Cohn.
So I guess the rationale to reduce the number of doses so that travelers can get them in doesn't entirely, we do have other traveler vaccine recommendations where if you are partially vaccinated, you go ahead and go and you get the final dose when you come back. So persons who would get the first 2 doses at 0 or 7 days would be more protected, but you could still, I guess I would caution you to change the recommendation just so that travelers can complete the dose more quickly, but rather provide guidance for them.
Dr. Fry.
We weren't changing the schedule because of that specific thing. It was just a consideration. Yeah, if they need more, we would not solely recommend that.
Are there other comments or questions? Dr. Lee.
Just really briefly to summarize, I think some of the concerns, it seems like a better understanding of the implementation considerations might be useful here, and I'm thinking around adherence to the ability to do serologic testing, as well as potentially getting a booster dose. And we were just thinking about potentially private practitioners for veterinarians. I'm assuming that, if you're a part of a health system or a clinic, or you're part of animal control, that there would be more routine sort of regulatory standards in a way, for lack of a better term, but just for private practices that may or may not have that same level of oversight, just to make sure that we're enhancing protection for the population. It may be a very small proportion. I just don't know what the distribution of work looks like for the veterinary population.
Dr. Bell.
And I guess I know that you've presented some information about the rationale for this change, but I guess I would like to understand that a little bit more, because I think in my mind I have a certain amount of this, if it's not broke, don't fix it feeling about all of this. And so I'd like to understand the rationale better.
Dr. Paling.
And then following up on a comment that you made earlier, it would be very helpful if we could figure out how many of the doses administered of the 15,000 are in which of these categories, and that would also help with the thought process.
Any other comments, questions? Dr. Fry.
I was just wondering if the public health people in our circle here would have anything to say about what happens in their practice or what their concerns are.
I haven't worked with our rabies team recently, but most of the work that we do is in a post-exposure prophylactic setting, and so I don't think that we really can speak to the pre-exposure.
Is there anyone from the company here who could perhaps speak to anything they know about where the doses are distributed to? Is there?
Anyone else?
Dr. Friedland, are you coming or no? It's up to you.
Hi, Julian Ritchie, Sanofi Pasteur. I was trying to signal you. I don't really have any specific data to be able to provide you as far as distribution. We can certainly look into that and come back with that.
That would be great. Thank you.
And Leonard Friedler from GlaxoSmithKline, the exact same answer, and we'll get back to you with that information.
Thank you.
Sorry to make you get up.
Yes, I didn't see you. Forgive me. Go ahead.
Hi, this is Chris Finley from AIM. I will say that in our area, we're aware that vet technicians are often not covered and that they actually do private clinics where they do intradermal 3 times to try and meet it because it's 1/3 the price. And so cost becomes a real issue. And we hear about how they set it up. They know it's not ACIP recommended, but it's the best thing they can do to be protected. So the idea that it's occupationally covered doesn't always work.
Thank you for that information.
So maintaining a 3-dose series in that case versus a 2-dose series, I don't know if a 2-dose series would be accepted better by that group and potentially would take it intramuscularly instead of risking the intradermal route, but potential consideration.
Anybody else with questions, comments? Please go ahead, CST.
Yeah, Chris Hahn, CSTE. I'll just mention also anecdotally, in Idaho at least, we often have, exposures in veterinary offices, of course, they're treating an ill dog or ill cat. And I'm surprised how often the staff is not vaccinated. And I do think cost is probably part of that consideration. I would have assumed that was just very routine. It doesn't appear to be the case, at least in our state where we don't have terrestrial rabies.
Thank you. Anyone else? All right, then I think we'll draw this session to a close.